again, everyone. Good afternoon. I hope you all had a great lunchtime. It's a pleasure to be here once more in my third edition as a GVENC Scientific Committee member. My name is João Paulo, and I am PhD student in Genetics and Plant Breeding, supervised by Professor José Baldim Pinheiro. Now, I would like to invite you to your first roundtable of the 9th EMPB, entitled Bring Together Genes, Phenotypes, and Environment, Integrate Strategies for Prediction and Selection in the Agriculture of the Future. This roundtable will feature three distinguished speakers, Dr. Katherine Fraus, Dr. Evandro Novaes, and Dr. Alexandre. Your first speaker is Dr. Katherine Fraus, Assistant Professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln. Dr. Fraus earned a bachelor's degree in agronomy, specializing in plant breeding for Iowa State University, and a PhD in agronomy and horticulture, specializing in plant breeding and genetics from the University of Nebraska-Lincoln. She leads the state's small grains breeding program, working on the genetic improvement of winter wheat, barley, and triticale, focus on yield, quality, disease resistance, and sustainability. Dr. Fraus integrates advanced genomics and phenomic tools in multi-environment trials to study genotype by environment interactions, especially across rainfall gradients. Today, Dr. Fraus will present her lecture entitled Identify the Small Grains Varieties of the Future with Genotype, Phenotype, and Environment Information. Please welcome Professor Dr. Katherine Fraus. The audience is all yours.
All right, as you can see, I somewhat changed my title to kind of reflect more of what the story. It is still the same topic. This happens to be a new problem for our breeding program, a pest issue. And so I'm going to tell a story today about genomic and phenomic research and how we're trying to implement it in the newest part of our small grains breeding program. So, this pest. Wheat stem sawfly is a native North American pest, infests native grasses, moved into spring wheat, and eventually moved into winter wheat production in Montana, and then has slowly been making its way south. And it has arrived in Nebraska, started appearing in the late 1990s, really became a problem around 2015. This pest actually is a little wasp. It lays an egg, a single egg in the stem, and then throughout the growing, so it's laying that egg right around flowering. Throughout the rest of the grain filling period, that larva is slowly eating its way through the stem, crawling down to the bottom, and then right as the plant begins to senesce, we end up with that larva creating a overwintering home, and then it actually saws, hence the name sawfly, cuts off the stem at the bottom. And this is where we get a lot of the economic damage from this pest. So this is a nursery that has been severely infested with wheat stem sawfly. And as you can see, that is unharvestable. So 20 to 30% yield loss in an average farmer's field. And we also see effects on the loss of photosynthetic capacity because there is a larva chewing through that stem. And we see requisite effects on the grain quality and the seed size in infested wheat stems. So this is really challenging because this pest is really not visible to the naked eye. There are no physical symptoms that we can see until it makes that cut. So if we want to phenotype this, this is really quite challenging. We do have some genetic resistance methodologies. The main one is breeding for solid stem wheat. So as you can see from this 1 through 5 scale, most wheat is like a straw, it's hollow. But if we breed for more solid stems, we have resistance to that pest, and the larvae do not successfully make it all the way down to the bottom to make that cut. We have a major gene for this. Yay, fantastic. This should be so easy. The reason I'm telling you this story is because it's not easy. The presence of this marker, and we have a very good Cas marker for this. It's really simple to run, it works great. It doesn't match the phenotypes all the time. So it is suggestive that it's probably solid at some point in the life cycle of that genotype. However, the stem solidness is very impacted by both phenological and environmental effects. So some varieties with this gene may be solid at earlier before flowering and then become less solid after flowering, so that's not gonna help us very much. If it is a cooler, cloudier, wetter environment, then we have a lot less solidness. So absolute pain in the butt trait to measure. As you can see, has to be destructive measurements. We also want to look at other resistance mechanisms. So antibiosis, meaning the plants might actually kill the larva, or non-preference, meaning that the sawfly don't want to lay their eggs in that genotype. However, that requires, again, destructive phenotyping. We actually have to slice open those stems and see if there are larvae in there. So you can see what that little larva looks like. Stem cutting can only be evaluated at the end of the season if that's going to be our phenotype of interest. And if we want to see significant yield cutting or stem cutting, we don't get yield on those plots because by the time we have bad stem cutting, they're unharvestable. So we have to make a decision on what kind of data we want. So the other thing that I didn't mention is my breeding program is based in eastern Nebraska. So I'm based at our main campus. This pest is only in our main production region, about 5 to 6 hours away from Lincoln. So what do? We don't have this pest where it's close by. We go harvest a bunch of bundles and pack them into a minivan and drive them back east. And then we have interns. Actually, we've got one of our Brazilian interns who's here today in one of these pictures. And then we have students either split horizontally or split vertically, depending on which phenotype we want. So this is really time consuming. If we were to have one person on this, it would take over 4 weeks of work to phenotype all of our lines and thousands of dollars in salary. For reference, my turnaround time from harvest to my planting season is 6 weeks. Don't really have time for that. So we wanted to see how can we optimize our breeding program, how can we move faster, make better selections with less labor. Now, I'm happy to talk later. I don't have a lot of the methodology and the details about how we modeled these and all of the different genomic selection models we've tried. We can talk about that more. This is just kind of the overall overview of this project. So what's better than having to slice into stems so we can get empirical phenotypes for genomic selections? Let's take pictures. So that's what we tried first. What we have here is a phenomic selection model where we have, and I feel like I'm missing a slide that hopefully it'll come up later, that says when we collected this data and what we collected it with. But if not, I'll try to make that up as I go along. So we divided we had flights over the growing season at 2 different locations in Nebraska. Divide that up through cross-validation to develop our phenomic selection model, and then apply our testing data set. We use both cross-validation and leave one environment out. Phenomic selection has worked really well in certain crops, even in wheat for certain diseases. However, they are primarily visual traits, so we've got a bit of a reach here already. But remote sensing has worked well for traits that are invisible, like water stress, nitrogen deficiency. So it could work. That's what we thought, right? Here, this is what I was hoping was earlier. So the study that we did this in, you're going to notice that this is a really low amount of genotypes because this is in kind of the beginning of our breeding program. We don't have a lot of lines yet, and also where we have more lines in our early generation trials, those are located in Lincoln, in that eastern part of the state where we don't have the pest. So our elite yield trials is what we have in the presence of this pest. And so we've got kind of limited entries and 3 years. Well, for the phenomic selection, we only had 2 years of data. We did also genotype these with a 3K SNP chip. And so in total, we have 5 year by location environments. All right. So our traits, from all of those bundles of wheat, we went through and selected 10 samples. Ideally, we would want to do a lot more than 10. But again, we're working against time and level of effort. So because you can't rate both infestation and stem solidness, we were limited to rating stem solidness at just the bottom node, because once you split that vertically, it's very hard to estimate what the level of solidness was at all of the inner nodes. So we just rated solidness at the bottom of the inner node, and then we looked at infestation, where we split that vertically and looked for the presence or absence of the larva. Also got yield with a Zern plot combine. Little about our drone. We ended up with 8 vegetation indices and 4 spectral bands that we were attempting to correlate with these sawfly-related traits. Here's our leave one out and our 70-30 cross-validation. All right, this is probably where we should have just quit, because we look at these correlations and level of significance. We did have significance for infestation based on genotype, location, and year, no interactions. Stem solidness actually was pretty good for significance. Heritability on both of those, okay. Our yields, because these were selected for solid stem traits, not for yield, and we had some really challenging environments in this set of years. We actually had really low heritability. I don't know if you've ever managed to get that low of heritability in your study, but I get the prize there. And it really comes down to those interaction effects. So you're not going to be able to see a lot here, but the important thing to note is that you do not see a lot of red on here. Red are positive correlations between a vegetation index or spectral band and the stem solidness trait. So this happened at various time points. So it looks like the only significant ones we had were in Hemingford in 2024 at grain fill, and then Sydney 2024 at grain fill, but not consistent vegetation indices or bands. So no consistency over the 2 years. Looking at our infestation results, again, you don't see a lot of red on there. This really is not working well. With our correlations, we went ahead and looked at prediction accuracy. Turns out, even with that low heritability on yield, we did better at predicting yield than we did either solidness or wheat sawfly infestation. So And if we start looking at our leave one out environment, we don't have any consistency over environment. So, bummer. Looks like we're not going to have a lot of success with our phenomic prediction. It was worth a shot, right? A lot easier to fly a drone than to do this. I will say we do have some caveats. This is a population that's fairly limited in genetic diversity because our breeding program is still small for this trait at this time. However, literally every solid stem wheat out there comes from a single source. So especially when we're talking about the solid stem trait, there's not a lot of genetic diversity in wheat in general. So our conclusion here, phenomic prediction is probably not going to work. This paper is in review, and some of our reviewers argued that, no, you can't say that, your data set is too limited. And we're like, it really didn't work, guys. So I'm pretty confident in saying, at least with a multispectral UAS system, this is really not going to work for this particular pest. So let's think about genomic selection. We've seen posters, we've had some talks looking at this already. Goal is to do that same level of prediction, but with genetics, right? However, we still have to have those empirical phenotypes, which is what we were hoping to avoid with the phenomic selection. Here we did look at single trait models, Bayes A, Bayes B, GBLUP. We also looked at combining wheat stems, the infestation and the solidness, and looking at both single trait models as well as our multi-trait models. So this is all implemented in R and utilizing a fixed environmental effect. So just as a reminder, when we're talking about those multi-trait genomic selections, this is working best when the traits have a high genetic correlation and a highly heritable trait. So we're, okay with our traits. Has worked well, and I think the important thing to remember here is the trait we're trying to predict is that focal trait. The other traits are basically supporting information for that prediction model. Just like our phenomic prediction, we have a cross-validation testing method as well as a leave one out environmental model. So, like most genomic selection studies, not a lot of difference based on the model when we're looking at our cross-fold validation. We predicted solidness at a moderate level, infestation really well, which I find interesting, and I don't know if I necessarily trust that. Is it just because we have a lot of sawfly out there? I think that's an area of further study for us. And then stem cutting, which is something we would really like to predict because we can't get that trait without sacrificing our yield data. We again, pretty moderate prediction levels. When we're looking at that multi-trait model, or the leave one out, single versus the multi-trait, we really see this is where it actually maybe matters that we have that SST1 single marker data, because that Bayes B model actually takes the major genes into account. We do see improvement of our prediction models if we have that gene under consideration. So despite me being kind of a downer on the SST1 marker, it does actually work probably better in a genomic selection model than it does when we're just trying to use it as marker-assisted selection alone. But again, we don't see a lot of strong conclusions here other than, or different environments, other than, that Bayes B model is probably better than the multi-trait models. And that's pretty much what I just said. What we came down to is that our multi-trait models didn't perform as well as we hope, likely because our genetic correlations between stem solidness and yield aren't that great, and softfly infestation and yield aren't that great. Again, this is probably where more data could really help us. I don't think more data is going to improve our phenomic selection results, but we are continuing to improve this model, adding more genotypes as our breeding program for this trait grows, and we have more genotypes, as well as, unfortunately for my growers, softfly has moved eastern in Nebraska, so we have gained another location that we can use as an environment. So not only are we adding more genotypes to this genomic selection model, more data, we can also add more environments. And so that is just going to help these predictions improve. And this is where one of our future objectives is really to think about how can we deploy a sparse testing model, because the way we've been doing this is we are doing that stem sampling and stem dissection on every genotype at every location we can. It's a lot of work, it's a lot of hours, it's also a lot of driving. So if we can deploy more of a sparse testing model, can we manage to leverage the same amount of information with less work? So that's really a future area of study. So conclusions for this. Unfortunately, internomics are where we're still at for this wheat stem softfly trait. We cannot replace that empirical stem dissection with a UAV. Genomic selection is probably going to be the best way for us to improve both yield as well as the, not necessarily infestation, but reducing the level of damage that the softfly causes. We don't really care how solid they are, as long as those wheat varieties are solid at the right time. And that can be very difficult to phenotype, because we would have to be out there dissecting stems multiple times in a single plot. So genomic selection is probably helping us get around that a little bit. Like I said, we can predict wheat stem softfly infestation pretty accurately, but is that just because there's a lot of softfly out there? And hopefully, as we get more data, so we now have three, one on-farm location with a producer and 2 university farm locations that are severely affected by this pest, so we'll have a lot more data coming in. And so where I want to end this is I want those of you who might be interested in an internship, forget what I said about internomics. But we do take interns every year. We have a few slots left for 2026. We do everything from 3-month internships to a year. University of Nebraska has been great to work with international students, and we have a lot from Brazil. So I have and I will say I forgot to check and make sure that this link is still working. But you all have my contact information, so if for some reason this application form is not working, let me know, and we'll get that fixed. But we are a breeding program. You get a hands-on training in breeding program logistics, data collection, data analysis, and so we always love to have a new crew every year. So that's where I'm going to end, and a little bit shorter than the time you gave me, but that's great because we'll be back on track after our long lunch.
Thank you very much, Professor Katherine, for your insightful lecture and sharing your valuable knowledge with us. And now, to continue your roundtable discussion, I would like to introduce your next speaker, Dr. Evandro Novaes, professor at the Federal University of Lavras, Brazil. Dr. Novaes earned a bachelor's degree in forest engineer from Exalc USP, a master's in genetic and plant breeding from the Federal University of Viçosa, and a PhD in forest research and conservation from University of Florida. He leads the forest genetics and the breeding group called Gen4 at UFLA and teaches courses on genetics in agriculture, population genetics, and genomics and bioinformatics. His research focuses on forest genetics and breeding, especially eucalyptus and exotic hardwood species, as well as molecular tools for forest genomics. Today, Dr. Novaes will present integrating molecular and environmental markers to predict complex traits in plant breeding. Please, Professor Evandro Novaes, welcome. The floor is yours.
All right. Little bit nervous. It's an honor to be here. So he told already about my title. Sorry about my English. It's a little bit rusty, but let's go. So for every breeder or geneticist, of course you know that the most economical important traits you work with are quantitative. That's not different in forest, you know. If you look at wood growth, wood quality, resistant to many abiotic and biotic stress, they are all quantitative. And that poses a challenge because these traits are very much influenced by environment. And worse than that, the way the environment influences, it depends on the genotype. So here is a study that we conducted when I was a professor at UFG in Goiânia. You know, Goiás State, right at the central part of Brazil, around Brasília, our federal district. And we brought there 113 clones of eucalyptus, and we planted in three different municipalities or regions, very close by if you think about Brazil as a whole. Even though they were relatively close by, you see what you have here is the wood productivity. Each point is a clone, and the lines connect the productivity of each clone across the three regions. And as you can see here, unfortunately, it's a little bit difficult to see, but some clones that have average performance in one site becomes top 4 or top 3. And that's a big challenge for breeders because you have to conduct many, many tests across the target population of environments that you want to plant. So how can we handle this problem? You know, there are many traditional strategies. Of course, you allocate your tests throughout your regions that you are interested in planting, and then, like here, you know, you can select the most adapted clones or genotypes, hybrids that you have. I would argue that that's quite a risky way of going, you know, because the past 5 or 7 years that these eucalyptus were planted, the environmental factors at the past will not be the same in the future. You know, we are really selecting plants to plant in the future. You know, in the next 7 years won't rain the same, won't rain on the same days. The temperature will be different. So I would argue, even though, you know, for students that may be a really way to go, there's, of course, stability. There are many how do I say, many definitions for stability. But I won't have time to discuss that. But it suffices to say that, you know, it's better maybe to select clones that are pretty good, have pretty good yield, but have stable. In other words, they perform well in every single or most environments. It's a little bit riskier to do that. And I'm a big believer that agriculture, and especially in forests, we should care more about diversity, you know. The way forest companies go nowadays is to plant a single clone in an entire field. You know, thousands of hectares with a single clone, which is the monoclonal strategy. On this article, people from, which is now Suzano, Gabriel Deon, it's not in Suzano anymore, and Magno, my past colleague who retired at UFLA, they showed on this study, you know, the concept of clonal composites, which you, instead of planting a single clone, you select 6, 10 clones that perform reasonably well, and they grow about the same, at the same rate, you know, so they don't compete very much with each other. And what they show on this paper is that you not only have more stability of the productivity, you can also grow more. You improve the productivity on this paper. And Gabriel Deon used to explain this with an analogy of sports. You know, the monoclonals is like a tennis match. You know, if Rafael Nadal is not doing great that day, he will lose the match. On the other hand, as you can see, I'm old. I used to see basketball when these guys were playing. And when these guys were playing, even when Michael Jordan was not doing well, they could still win. You know, Scott Pippen and Dennis Rodman, they could pitch in and win the match. That's the same thing. Clonal composites works like that. You know, if you have a problem with one clone, you still have 6, 7 more to pitch in. And you know how forests compete. You know, if one is not doing well, the other one will compensate for that, because this lower performance cloning is not competing well, so the other one will pick it up. So, but what causes GBA interaction? This is a study, an old study that I did on my PhD under Matthias Skirst's lab, and we showed, you know, that a single nutrient, nitrogen, can completely change the control of quantitative traits. We did a QTL study with a single population. It's a poplar population. So we crossed 2 species of poplar, got this hybrid population of about 400 genotypes replicated with 6 clonal replicates to make a study that tested 2 nitrogen treatments with 3 biological replicates. In other words, same population, same site, same greenhouse. The only difference here is one nutrient, nitrogen. Of course, it's a big nutrient, right? It's the most important. But anyway, we phenotyped these 400 genotypes for biomass growth, food quality. We also did a genotyping of microsatellites to perform a genetic mapping with the markers, and then to correlate the genetic variation with the phenotypic variation in a QTL analysis. But what I want to show is the main result here. Here, each bar is a QTL for a specific trait, and what you have on the right side of each linkage group, which represents a chromosome, are the QTLs found under high nitrogen. And what you have on the left side are the QTLs identified only with under low nitrogen. And I want to emphasize again, it's the same population, same field, same greenhouse, the only change is a single nutrient. And as you can see here, you completely change the genetic control of the trait, a single nutrient. Imagine if you plant the soybean here in Piracicaba, and you go 50 kilometers going to Campinas, and you plant. It's not only a single nutrient that changes, everything changes from field to field. Maybe on the same farm, you know, and that complicates things a lot. So what causes GBA interaction? When you change a single nutrient, when you change field, you change the gene expression of genes. Therefore, the genes that are important in one site or under high nitrogen are not the same genes that are being important under low nitrogen or on another site. So it's very challenging. And I want to argue that we breeders, of course, we are geneticists. We focus on the G part of the component of the phenotypic variation. What I want to argue today is that we also should look at the E part, the environmental part, because as I showed, it is very important. And it's actually, I'm going to show, more important, unfortunately for us geneticists, but we should speak that on a low tone, you know, because otherwise the guys from the agronomy department will think they're too important and we are not. Anyhow, so the idea is, as Bruno from Rafael Tassinari showed yesterday, is the same thing. Rafael Tassinari is doing the same thing that I'm going to show here. It's for us to characterize our fields, maize fields or eucalyptus fields, with environmental covariates. Geographic covariates, altitude, longitude, soil covariates, clay, sand, soil pH, cation exchange, and so on, and also precipitation and temperature. You know, why is that? For us to improve modeling of the G by E interaction, because if we understand the G by E interaction, if we model it, we can predict, you know, how is my clone going to produce in Lavras, where I work, or in Goiás State, and so on. That's the idea. And this is one brilliant student I have. This is his master's article, Leonardo. He's now working at Syngenta. Even though he did not finish his PhD, he's still doing his PhD, but the company already took him from Lavras. Anyway, this is published on the TGG journal, and it's entitled Prediction and Mapping of the Productivity of Eucalyptus Clones with Environmental Covariates. So this is a partnership with Suzano. We contact them with this idea. We asked, which are the most planted clones you have there? And we wanna, can you give us the productivity data you have? Because, you know, in forests, the companies monitor the growth of the forest so they can plan which field they're gonna harvest today, because the industry has to work, has to run 24/7, 300, the entire year. So here are the regions of Brazil that Suzano has farms and also industries: São Paulo State, Mato Grosso do Sul, south of Bahia, north of Espírito Santo, Maranhão, Pará, Tocantins. And they gave us more than 13 productivity data from operational fields. So there's no experiment here, which may be a problem, right? And as it is operational data, we don't have all the 6 clones planted in every single region. So it complicates a little bit. But anyway, so we used all this productivity data from each of the 6 clones to try to model their productivity based on the environmental covariates. The nice thing about environmental covariates, you don't have to have a lot of money, you know. The data is already there on NASA Power, World Clean. You can download right now this data from anywhere in the world. That's what we did, you know. You have also soil grids, which is also a public database. I would say, I'm afraid I don't trust very much, but sometimes it's difficult for us to have 13,000 soil data from the company. I don't know if it would be difficult for them to give us. I don't know if they have a central database with all this, you know. And also the geographic data, latitude, altitude, longitude, which is already available with the sites. So the way we model, there are several ways of modeling this environmental data. On Leonardo's master dissertation, we used the partial least square regression, which works similarly to principal component analysis, but it's more focused on the interaction. Then we tested the validity or the predictive ability of our model with cross-validation. Having detected that works somehow, we can now then predict the productivity of every single clone at every single pixel in the map. That's the nice thing. Since we have the productivity data from NASA Power, or in this case, World Clean, at every 50 kilometers square from Brazil, we can predict. Of course, you cannot extrapolate much. You know, if we did the modeling on those states, I cannot, you know, predict on Manaus state, for example. So you have to define your, as Bruno showed yesterday, your target population of environments, which cannot be a lot of extrapolation. I forgot to turn on my timer, anyhow. Katherine, 15? But Katherine gave me 10 more, right? I'm kidding, guys. And then, of course, you can recommend the clones or the cultivars at the pixel level, you know, which is another big increase. And you can also, sorry for the Portuguese here, it's a mixture, but anyway, you can use the environmental covariates to estimate the environmental similarities, you know, between all those four regions of Brazil. And what we detected, which is not very unexpected, is that, you know, there is a big correlation between the distance between the 4 locations and the environmental similarity. The farther apart, with every increasing of 100 kilometers between 2 states, you decrease their similarity in point 22. So how does the model work? This is the predictive ability of the model. What you have on the x-axis are the predicted values and the observed values on the y-axis. And you can see the correlation is somehow good for most of the clones. You know, here's for clone 1, 2, 3, up until 6. Of course, correlation is very influenced by extreme values. So I don't trust very much this correlation here. This correlation is not good for this clone 3. And also not for this clone 5 also. These 5 points here are influencing this high correlation. And another thing I want to argue is, if we use environmental covariates, we can recommend our cultivars in a much better way in the future. Because now we know which are the environmental covariates that influence each of our clones or maize hybrids. And as expected, because of the G-by interaction, the environmental covariates that influence clone 1 are not the same that influence clone 6, for instance. So the idea here is, if you identify the most important environmental covariates for each cultivar, you can not only recommend the clone, you say, nowadays we say, oh, plant this clone here in Piracicaba, there in Campinas, but don't plant in Mato Grosso do Sul. Now we can say more. Plant in Piracicaba, Campinas, but be aware, this clone does not tolerate high altitudes, for example. Don't plant under high altitudes. So it's a much. So now we breeders, we are getting closer to the agronomy department, for example, you know. So if we trust the model, we can now predict the productivity of every single clone at every pixel of the map. And each pixel is defined, the resolution is defined based on the resolution of the environmental covariates we obtain. So if we obtain a very high resolution, 5 kilometers square, you can predict F at 5 kilometers square. In this case, our prediction is about 50 kilometers square resolution. No? For every single clone, we have the prediction under this target population of environments. As you can see, we did not extrapolate much because that would be a problem, right? And you can now compare which are the more productive clones at every single pixel. And then you perform a recommendation of cultivar at a much higher resolution, right? And this is the map. Each color here now is the color of a clone. For example, in Mato Grosso do Sul, our model recommended clone 1, which is actually the most planted clone there nowadays, and clone 6, and so on and so forth for every single region, for every single of the 4 regions we did our study. This is another study, very similar, but now with families, not with clones, with progeny tests. This is work done in partnership with Paulo Henrique da Silva, which works right here at IPF, which is the Institute of Forest Research. Very nice guy, very competent, and a friend. And also Leonardo, this is his work for his PhD project. And this is, I entitled Predicting and Mapping Eucalyptus urophylla Family Performance Using Large-Scale Environmental Covariates. And we also have some genotyping, SNP genotyping here. I'm going to show you guys. The nice thing about IPF is it unites all the bigger companies in Brazil, you know, forest, pulp and paper companies, what else? Energy companies, the coal, they use, you know, the metal industry. Anyway, so they can plant, you know, this progeny test all across Brazil, from Rio Grande do Sul up until Amapá, in the main regions of eucalyptus plantations across Brazil. So we have 18 progeny trials in each of these dots, each of these states, with 107 open pollinated families of Eucalyptus urophylla. Very large study. We now collected environmental data for every single of these 18. You can do, as I showed before, the environmental similarities among this. We can model with different models the productivity now of the families, not of the clones. Now I'm trying to predict the productivity of the families, test the predictive ability of each of these models, define your target population of environment, again, avoiding extrapolations, and then you can predict the productivity of each family at each pixel of the target population of environments and make a recommendation at the pixel level also, the same idea. So in this case, Leonardo used 546 environmental covariates. So he did many combinations, quartiles, median, mean of daily temperature, precipitation, monthly, and so on and so forth. And as you can see here, we can build this W matrix, which is the matrix of environmental similarities among the 18 sites of Brazil. As you can see, you know, these sites that are here in the southeastern part of Brazil are way more similar among them than the 2 sites up north, which are Amapá and Maranhão. They are not similar at all with any of these. Also, Rio Grande do Sul and Paraná are also very different from the others. In this case, we collected data, I forgot to say, from NASA Power and soil grids. And you can also group the environments, you know. Those right here are this blue group, Rio Grande do Sul in black here and there, and the 2 up north here, and the 2 at Bahia here. So you can see that these environments way south or way north are very different from the main region. And we also, as I said, genotype these families. But we are at the university, we don't have a lot of money, so we try to genotype in a clever way. So how did we do? We selected 24 individuals from each progeny, and we extracted the DNA in bulk, and we genotyped with an Illumina SNP chip from eucalyptus, but we use the bulk DNA. You know, traditionally, when you use the genotyping SNP chip, you get for each plant and each SNP, you have either a 0, 1, or 2. 0 if it is homozygote for one allele, 1 for heterozygote, and 2 for the other allele. And now here the idea is for us to try to estimate the allele frequency of one of the 2 alleles, right, in each family. And this allele frequency now goes from 0 up until 1. 100% is when you are homozygous for that allele, and 0 if you don't have that allele at all. In other words, you are homozygous for, in other words, the family don't have that allele. I'm being confusing here. Anyway, so what you have here, instead of 0, 1, and 2, you are going to have, as I said, values from 0, 0 to 1. And now you can use the same strategy, a GPLUP that is used with the SNP genotyping, but you can now use it to estimate the relationship between families, but now with allele frequency. And we tested, we opened the book for 3 families. Family 74, 92, and 109. What we did, we genotyped we genotyped the book and also every single of the 24 individuals for each of these families. And the results are quite good, you know. The correlation between the frequency for this family is estimated based on the individual genotype and based on the book are at 0.96, very high correlation. We also did a principal component analysis of the individuals from each family, each of these 3 families, which is like the diamond here, and the book, the frequency, the allele frequency are the dots. And you can see that the allele frequency are at the middle, about the middle of the group, and they are distant from each other. So we tested, let me go a little bit further here. We tested 5 models, right? The traditional model without marker, without genetic marker, and without the environmental covariates. So you have the sites, we have the family, and the G by E. The small g here is when you estimate the genotype, the genetic value with the markers. And the W is when you use the environmental covariates. So how was the phenotypic variation partition among the site, the W matrix, which is the environmental covariate matrix, and the G, which is the genotype, either evaluated with markers or with only the phenotype. And as you can see, since we are all across Brazil, the environmental variance is very important, as I said, you know. Don't tell the agronomy department, not the irrigation people, don't tell them. Let's keep that among us. But as you can see here, 80-something% of the variation is environmental. The genotypic effect is here in red, and the G by E either evaluated with environmental covariates in brown, or with the sites here in orange are there, right? So now let's test the predictive ability of each of these models in terms of sparse missing data, CV2, predicting missing genotypes, entire genotypes, and predicting new environments never tested, and also the most difficult scenario, which is the CV00, is for you to predict unevaluated genotypes on unevaluated environments. As you can see here, this is very challenging. I think other people already showed. It's the CV00 is at 0.19, even with markers, molecular markers, and environmental markers. But let me show a little bit more. But as you can see, you know, the environmental covariates help significantly, I would say, improve a lot the prediction ability of the new environments. And also we have a pretty good predictive ability of new genotypes. But, you know, I think this is a little bit inflated because we have a lot of environmental variation. So the environmental variation is counting way more here, right? And anyway, so if we can look a little bit at more details, the prediction of new environments with each of these 5 models. So each bar, each section here is one of the regions, you know. RS is Rio Grande do Sul, Here is Amapá, and here is Maranhão, the two most distant, most northern states. And the predictive ability for Model 1 is the first bar, Model 2 the second, and so on. As you can see, for Amapá, which we know is a very challenging environment for eucalyptus, very, very high temperature, a lot of drought, and our model doesn't work at all. It's negative predictive ability. The correlation is negative. But some things are very interesting. Remember that Rio Grande do Sul was very different from these other sites? But besides that, the use of environmental covariates did a big improvement for Rio Grande do Sul, big improvement. Mato Grosso do Sul, which is right here, was very similar to the São Paulo, Minas Gerais sites. But for some reason, the environmental model improved, of course, but still, the predictive ability is way below the other sites around this region. This is all to say that we have a lot to learn out of this, out of the environmental covariates using. There's still a lot of research to be done, and we are just starting, I would say, right? And of course, as I showed before, for the clones, we can also recommend the families at the pixel level. But anyway, I wanna stop. Do I need to use the 10 minutes? Acabou? 5 minutes? Okay. So I want to argue to finish that, predicting, even though it's very difficult to predict G-by interaction, this is at the heart of the most important problems in genetics, I would say. Either, medicine or plant breeding, when we need to predict, for example, we need to have models, where we can look at an eucalyptus seedling and somehow be able to predict if it's going to be a good plant in the field or not. But as I showed, it's very difficult, 0.2 CV00. But it's surprisingly difficult, because if we look at a clonal forest, for us that are forests, every single tree grows about the same. The crown, the diameter, they're all the same. They look like identical twins, On the other hand, when you look at that seedling forest grown from seeds, every single tree are different. Every single tree are different, just like fraternal twins or brothers and sisters. So how come this, when you have identical twins, they look so much alike, clones are so much alike in the field, you may say, because these clones are planted in the same environment. Identical twins share not only the genetic, they have the same genetics, but they share the family also. They go to the same physicians, they eat the same thing, they have the same culture, and so on. But what if I show you these cases of identical twins separated at birth? All these 3 pairs, they grew apart, completely apart. So this grew in, I forgot the state, in the US. They were both born in the US, but at birth, this other went to South Korea. She grew up in South Korea. Probably a very different diet, different physicians, everything was different in South Korea. But when they got together, as adults, they were very, very impressed on how similar they were. These 2 guys also grew apart, but they drive the same model of car. They smoke the same cigarette brand. Of course, we have the confirmation bias here. We will pay attention on the things that how do I say? Confirms our hypothesis. But anyhow, twins, clones, they show that genetics are important. And we have to focus a lot of our research on predicting these genetics components and also predicting the environmental action on genetics, right? So my final considerations are G-BI interaction, of course, you know, are the major complicator for breeders. Environmental covariates can improve modeling of G-BI as I showed, and they can also indicate management practices together with the recommendation. A geographic information system plus environmental covariates can improve the resolution of the recommendation, and for the younger geneticists here, we should continue to investigate, to do science to improve the GBI modeling, because this is a very important task. I want to thank all my past and current students, my collaborator, especially Paulo Henrique from IPEF, Suzano, and Sen Piquet, and I thank you all for your attention.
Thank you very much for your lecture, Dr. Novaes. Your talk was excellent. And you may have, so let's move on to your next speaker. And now, to continue your roundtable discussion, I would like to introduce your last distinguished speaker of this roundtable, Dr. Alexandre Hild Iono, postdoctoral research at Unicamp and currently at the Swedish University of Agricultural Science. Dr. Iono holds a bachelor degrees in Science and Technology and Computer Science from Unifesp, and a PhD in Genetics, Molecular Biology, Bioinformatics, and Plant Breeding from Unicamp, with a research stay at University of Edinburgh, UK. He works in bioinformatics, genomic, and plant breeding, with a focus on omics data, artificial intelligence, and machine learning. Dr. Iono has extensive experience in next-generation sequencing, metagenomics, proteomics, and data analysis of polyploid plants. Today, Dr. Iono will present the best of both worlds, integrate phenomics and genomics as a selective tool for plant breeding. Please welcome Dr. Iono. The floor is yours.
Thank you very much, João, and thank you very much for the organizing committee to have me here. It's a really pleasure to be. I'm really pleased to be here. Thank you. So today I'm going to share a bit with you the experience I've had in Sweden for the last year into developing ways of integrating genomics and phenomics as a selection tool, right? So as João introduced me, I have a very different background. So I'm not a breeder, I'm not a biologist. So I started my path as a computer scientist, but my PhD was in genetics and molecular biology. But nowadays, I started to transition from bioinformatics to more quantitative genetics approaches and focus into giving plant breeders more accurate and more explainable selection tools. So during my years on the PhD and also on the undergrad studies, I have worked and assisted a lot of researchers on several crops, right? Crops that encompass like grasses, forestry, fruits, with several differences regarding the genomic complexity, regarding the sizes of their genomes. But in all the projects that I have worked with and I have assisted, people should investigate those traits. What they all have in common is this aspect, right? So we are always interested in investigating them, considering their continuous distribution, and also modeling considering the normal distribution that they have, right? And we all know, since our basics on statistics, that the normal distribution is amazing. It has a lot of properties we can define with a mean and a variance. But for us, the problem is that the variance is splitted in different sources, right? So we can consider, for example, a phenotype as a sum of fixed effects, a sum of genetic effects, a sum of environment, also a sum of error. But as Evandro just presented us, we also have a part that is the combination of genetics and environment. So when we think about the phenotypic variance, we are thinking about a lot of components. And we currently know how to model all these components. It is a very well-established theory for that. So for example, we can assume for simplicity that the residuals have this characteristic of being independent and identically distributed. If we want to model the genetic effects, we can use molecular markers to build a genomic relationship matrix. If we have data from different environments, we can also construct an environmental relationship matrix. And also, if we start to combine these covariances, we can capture different effects and different interactions. But the main question is, what about image data? Where should we include the information on images into this well-established way of modeling? So the first point that we need to understand is that image data have a lot of different levels of complexity, which vary from the amount of data that we are going to have, the amount of information that we are going to have. So if we have, for example, a simple set of RGB images, how should we include this information into, for example, modeling yield? So we can start considering that the information retrieved from images represent a different trait, right? So we can define several digital traits. For example, plant height, coverage, which is very simple information that we can retrieve from drone imaging. And we can consider the difference of those measures across time, and use all of this as a new trait, right? But then we can also consider new traits as being a prediction from different modeling approaches. So instead, for example, instead of relying on plate height, we can use a deep neural network, get a prediction of a trait. Of course, this is not a network for plant science. And then we can use this prediction also as an additional trait, right? We can also think about the novel networks that have been developed and use the prediction also into the definition of the traits. So returning to the model, how would it look like? Instead of having a single trait, we will have a lot of traits. And then we have, instead of univariate normal distribution, we have a multivariate normal distribution with associated covariance structures, and a lot of information can capture by that. But is it beneficial for breeding? What is the impact of the long-term benefits that we can get from such an approach? So currently, my master's student employed a lot of simulations to investigate if using so it would really increase the genetic gains in the future. So what we have observed is that the only scenario where the genetic accuracy would be really worth it like would justify the inclusion of images in such a way would be if the additional trait has a very high genetic correlation, which is almost impossible considering that this correlation changes depending on the environment. So it was really disappointing for us. And the increases into the genetic gain itself was really low. So what can we do instead? We can think that instead of using this information as a trait, it can be, for example, a component of yield, right? So if I have, for example, plant high coverage, if I have chlorophyll content, if I have extra information, instead of considering this as traits, I can gather everything and considering as part of yield. And this can be really beneficial because then we can include many more traits than the previous approach where it's very limited to like some small set of traits. And we know that we have a lot of vegetation indices, we have a lot of information that we can retrieve from images, and it is really difficult to establish which one is the best. And also, if we think about neural networks, what about getting the information inside the network as a trait, right? Because in the end, a network will combine all the image information into an embedding. And this embedding has all the information that the image can give you. So what about using this as well for modeling the trait? So instead of considering Y as a multivariate distribution, we can add an additional kernel. So a covariance between genotypes, but instead of considering molecular markers, we can consider information from images. So this is something that we can use to couple all the information that we get from image, and it makes sense. So considering these points, I'm going to present you the results that we obtained on performing such approaches for potatoes. So these are some preliminary results that I obtained in my first year of my postdoc. And incorporating phenomics data into potato is really interesting in several ways. But the main one in Sweden, they already use drone imaging for monitoring potato growth, right? So images are very efficient as a way to identify abiotic stressors, biotic stressors, because the differences between plants are very easy to be captured by images. So potato is polyploid. I used to work with sugarcane. So potato is simple compared to sugarcane, but it still has some complexity, especially considering that it is highly heterozygotes. And the main problem on potatoes is that it is very susceptible to multiple diseases and it is clonally propagated. So it has its own challenges. And for investigating, like if it would be worth it to include phenomics on that, we performed an evaluation on 3 environments, considering a set of about 200 individuals. And I'm going to present you the results related to yield, right? But we also performed a lot of comparisons considering other traits, including nutritional ones. So the first approach, as everybody else presented, is evaluating the BLUPs to understand the heritabilities, estimating the blues for doing other analysis. And in addition to conventional phenotypes, we also obtained phenotypes based on image, right? So just some examples here of vegetation indices that we used with their corresponding heritability. If we combine everything together and perform a principal component analysis, it's really clear that the information retrieved from images can differentiate the environments in a very easy way. So this information you should save because we are going to use in some slides after. But if we consider the vegetation indices by themselves and start to perform correlations with different traits, conventional traits of plant breeding, the correlations are not very nice, right? So they are environment specific. The correlation between vegetation indices and potato yield are very small, usually not significant, and the main reason is because we are running drones, and potatoes are tubers. So it is really difficult to predict a tuber based on the leaves. It makes sense, right? But when we started evaluating models for doing phenomic prediction on potatoes, the results were really, really sad. Really, really sad. So we also incorporated SNPs. So this is a SNP array that they have established in the Nordic countries, and it is very good for the population they have. So we started to investigate how to combine everything into a model. So the first scenario, like the simplest one, would be considering as the trained data SNPs and images and phenotypes, traditional ones, and predicting the phenotypes considering SNPs and images, right? So this would be similar to a CV1 approach, and we can considering two different effects, one coming from genetics, one coming from the image information, and as expecting, combining both sources increase the accuracy for the three environments that we evaluated. But the main problem on such an approach is that for doing so, we need to plant. We need to let the genotypes grow. And this is not so interesting when we have molecular markers because we don't want to plant them. We want to get the DNA from seedlings and then save a lot of money into field trials. So, just another point that I forgot to mention. If we consider those, the components of variance from genetics and from image, it changes a lot depending on the environment and also depending on the trait. But it is interesting to see that it's kind of balanced if we consider like the contribution from genetics and the contribution from images, which means that in this prediction here, there is a lot of information that is not related to plant genetics, right? But anyway, what was like the most challenging aspect on this was, and the best application of it, plant breeders, they want to get predictions based on SNPs. If you need to plant, the money that you were going to save is not so interesting, right? So then I started to think about approaches of how to predict like the conventional phenotype based on SNPs, but considering the model trained on images as well. So this was like a bit chaotic for me in the beginning, because how can you use something trained on images to predict information that does not have image? But that's what the plant breeders were looking for. They want some magic. And let's return to that PCA that I mentioned to you before. What is the main point of images? What is the main contribution of them? Is the color, right? So nowadays, everybody that employs vegetation indices rely on colors. So if we think about all of the different possibilities of vegetation indices, we are measuring differences in colors. So differences in colors are also very good to measure senescence, many other characteristics. And here, in our data set, it was very clear that the information from image was really good on separating the environments. So, What I did was, if we have a component from genetics, if we have a component from the environment, and a component from their interaction, maybe it might work. So Evandro just showed us that using environment covariates is very beneficial. But the information from images can also be considered a way to measure the environment, and maybe even a better way to measure the management. So nowadays we are also interested on investigating E by G by M. So the models get a more big scale, right? So this kernel here, based on E, is constructed based on image, right? But only on the trained data. And then also, of course, one based on genetics and one based on their interaction. And these are the results we obtained. And it was really surprising to see that using solely the information from the image to characterize the environment presented the best accuracies that we could have observed. So this is really interesting because it means that for some cases in potato breeding, you should use phenomics as a pre-selection tool in order to decrease your data set in the first cycles. And after, when you have a reduced data set, you can start incorporating genomics to get good selections on different traits that are more specific to genetics, such as starch content and so on. But for yield, and especially for breeding programs that do not have a lot of resources, using this approach is very beneficial, right? So this is something that we called sparse testing based on phenomics. And we developed a methodology for performing sparse testing based on images instead of based on SNPs. So I got really happy with these results. I think you can imagine that my supervisor as well. And this is the direction that we are going nowadays, to not use a single source or the combination of both, but using each one in different steps of the breeding program in order to make it more economically viable, right? And more important than that, relying on simulations to investigate if the real benefits will be worth it or not. Because when we have the prediction accuracies, they tend to give us an impression that everything is amazing, that everything is working really well, but they are really optimistic. When we start to simulate and observe like the gains across the years, the scenario is completely different. So, in conclusion, what we have here is that combining both sources of information provides really good, not really good here because we are working with a very quantitative trait, but reasonable accuracies, right? And this combination can be really interesting if you want to investigate genotype and phenotype associations, because then we can get rid of some environmental part that you couldn't have got ridden before with the first step of modeling. And as a way of using sparse testing, phenomics seems to be a very interesting tool. So that's how I conclude my presentation. I would like first to acknowledge again the organizing committee and the funding agencies from Sweden, the university I am, the Swedish University of Agricultural Sciences, and I think now we are all open to questions. Thank you.
Thank you very much, Dr. Aono, for your excellent lecture. And now I would like to invite Professor Katherine and Professor Evandro Novaes to join Dr. Aono at the round table for your Q&A session. And now we have time for the questions for the first round table.
Okay, I have a single question for all of the three. The question would be like, in the predictor model, in genetics, in environmental interaction, genotype versus environment, I like very much the phenotypes, integration step. I also like very much of your talk, and I enjoy a lot your talk. They are wonderful talk. However, I missed at least a consideration about, as you're talking about interaction between environment and genotypes, I missed the epigenomic, epigenetic as a one component that could add something in your models. Could you elaborate a little bit about this?
So thank you very much, Professor. Yes, it is possible. The only problem that we have is the amount of data available and, of course, how to obtain this information, right? So I was discussing, on the interview, where is the girl? Oh, she's not here, but today we had an interview, and we were talking about it. The main problem on including other omics information and other sources of information is the cost. Right? So getting the information for all the population that we have and performing all the interactions would require a population of a very big size. Because when you start to play with covariance structures, the dimension gets huge. So if you don't have an enough size, then the estimates will not be good. And consequently, what you want to really investigate will be just like some statistical error. And this is not what we are interested about.
I don't really have anything to add to that. That I would answer the exact same way, especially in my case where we're working with a trait in the very beginning of its breeding history. I just don't even have that data. After the 2025 year, I now have an additional 3 environments of data. So it's just not really feasible for us to do that at this time.
Just thank you for your question. I have nothing to say. The only thing I want to say about this, for research purposes, I'm all in, but for the operational day-by-day of breeding, these are things that are very expensive, So environmental covariance, as I showed, is pretty much free. You have to have a computer and somebody. You pay somebody to get the data and analyze. Now, transcriptomics and metabolomics, those are expensive. So I don't know if it will be, feasible to incorporate on a breeding program.
Nowadays, but remember that SNP arrays was very expensive in the past also. And today, every single one has the SNP arrays in the genome. So maybe you can have something more, more robust in your models in the next years, I don't know.
And that's possible. The cost may decrease. Definitely, cost is something I mentioned in the interview earlier, why I don't incorporate that much. Transcriptomics stay in the research field. They don't go into the breeding program. But at the same time, the level of complexity for the analysis of transcriptomics versus a SNP chip, that's not feasible in the turnaround time that I have. So I make my decisions for advancement within 6 weeks. And yes, you can do some of that ahead of time and try to add in your data from the current year. But it's really hard to add new analyses into a 6-week turnaround time.
Thank you very much.
But Betch, there is also something really interesting to add what my colleagues just said. When we think about other omics sources, for example, transcriptomics, the main problem of us using transcriptomics as a way to get another kernel for our modeling approaches is that when we consider SNP data and we measure the difference between individuals, this is the genetics. So it won't change, right? If we have an enough number of SNPs and also individuals. But what is the difference between samples based on RNA sequencing data?
The same thing as in phenotypes.
Exactly. So it changes a lot depending on the environment, depending on who sampled, depending on which company, the bioinformatics. So it has a lot of factors that are very difficult to be used to create a kernel. So I do believe that it might be really useful for investigating complex genetic architectures, the inclusion of other omic sources. But as a prediction tool or something to be included into breeding best practices, I think it's a bit unfeasible, right?
Hi, I have a question for Evandro, but please, Alexandre and Katherine, feel free to answer as well. Evandro, usually when we are doing genomic selections, we are usually focused on predicting new genotype in a new year. And usually the moment that we do the predictions, we don't have the environmental data yet. So my question goes to the direction how we can leverage all this information from the environmental, trying to predict a year that we haven't seen yet. And usually most crop, the G by year is very strong.
It's a very good question. So I wish my student was here, Leo, because there are several on these databases, there are also databases predicting the future. I don't remember the name anymore, but the panel that predicts climate change, they have for every single pixel in the maps also the future. And now it's also very important for us, breeding is getting very complicated. I kid my students, when I was doing my undergrad here, I hated, climatology. I did 5 soil science courses and, I like genetics. But now everything is coming together, and, is it going to be a year with El Niño or La Niña? That's going to impact the climate of next year. Being short now, you have ways to predict the future climate with some uncertainty. And I think for us breeders, it's a very hard task, but maybe the guys from the climatology department know better than us about that.
So I have a question also to Dr. Evandro. I know that the main focus of the event is on crops, but I have worked also with conservation genetics and things like that. So we have been using genomic in conservation genetics to try to conserve natural populations and things like that. How do you think that we could integrate environmics and G by E to select some natural populations, some individuals that's more suitable to some target environment to conserve that population or improve the chance of that population to thrive, to still exist?
Sure. I think the way to go, at least on the way I see right now, with your question is, you can model the distribution of the species you are working with. It has a distribution, so you can use this distribution with the environmental covariates to model its adaptability. And then now you can use, going back to his question is, how is it going to change now? What are the main sites that's going to be adapted in the future using the environmental for the future? We have a paper that we are about to publish. It's accepted that we model the Tona ciliata is cedro australiano, Australian cedar. We model its distribution, its adaptability across Brazil using the Australian zone where it occurs. You got the environmental data from Australia where it occurs, and it doesn't occur in Australia to model its adaptability. And then we now predicted where it goes, where it can be planted in Brazil nowadays, in 20 years from now, and in 50 years from now, using this environmental covariates with these future models.
Hey, I have a question for Alexandre. Do you think that maybe, you mentioned maybe the cost of phenomics, have you thought about using, for example, high-resolution satellite image, like WorldView, because I know the resolution might not be that high, but maybe combining it with genomics, it could something good can come out of that. I don't know. Have you thought about that?
Yeah, that's a very good point. I was talking to João yesterday about it as well. In the beginning, I tried it, and it didn't work. So in simple words, the idea is amazing, but the results are totally different. So it would be awesome, because you wouldn't have any cost at all. But then these areas that they use for the fields are very close by, and the way potato is grown, it didn't work. So maybe for other crops, this would be really interesting and can be used to replace all the imaging that we are using and build all those matrices. But on this specific scenario, it didn't work.
I'm sorry, and do you remember which satellite or which
No,
source
it was in the beginning. This was
okay.
how I started to go in this direction.
Okay, yeah. Thank you.
I can add to that, just that it has not worked for any of the breeders that I collaborate with. The scale of our plots versus a field scale it's just too small for the satellite capabilities that we have that are free. Now there are satellites with much higher resolutions that it's no longer free that might work. But I will say many of my agronomic colleagues who are studying more on a much larger scale the health of the crop or the type of the crop, what is out on the landscape, they have a lot of success utilizing those free Landsat, for example, even, or Modis. They've had pretty good success using those. It just hasn't quite come down to the breeding scale.
Yeah, it has to be private and expensive, yeah.
Yeah.
Still, yeah.
All the talks. My question is mainly for Evandro, but it's for everybody as well. Evandro, one data set that you presented that caught my attention was, I think, in the first or second slide, where you had that experiment that you did in a greenhouse where you only varied the level of nitrogen and you found like some QTLs control the traits and some not, depending on the level of nitrogen. And it got me thinking, because that is a great opportunity to investigate the causal link between nitrogen levels in the soil and the QTLs. And I would like to ask if you have any plans on trying to unravel why nitrogen is affecting those specific regions. Because, usually we only have observational data on soil, and that is an RCT with nitrogen, so it's an opportunity. I don't know if you explore that.
Yeah, it's a very good question. The thing about QTL mapping, especially with a very small population, like 400 individuals with 200 markers, you have a very low resolution. We have one specific region that controls pretty much almost all the traits: biomass, lignin content, cellulose content. We try to identify, which are the genes that control this QTL, but the region had 600 genes. So we had, for us to really fine mapping, we had to increase the population to 2,000 individuals, genotype many more markers, and it's very hard. But I want to add one thing to that, that's why QTL mapping never impacted breeding, because, the QTL that works under low nitrogen is not the same that impacts the trait under another nitrogen level. The QTL identified on this population, this cross, is not the same QTL on another cross. So, and it goes the same for GWAS also. Even though on GWAS you have so many different individuals, you don't work on populations, but you're gonna you identify very few genes. And so the thing is, quantitative traits are really, really complex. Hundreds, thousands of genes controlling, and you cannot identify those genes with such a small effect.
I think this is a point, kind of going back to the transcriptomics and understanding how the plant is reacting under different nitrogen regimes. Again, though, that goes to, you're probably going to be limited to studying that in a few specific genotypes rather than a whole panel or a whole breeding program, just because cost, as well as the complexity of the data. if you are trying to, I mean, if you're trying to make sense out of that, the huge levels of change in gene expression that are going to happen, adding more genotypes on top of that just adds to the complexity. And so it goes to how can you find the signal for the noise, and when you do find that signal, is it applicable outside of that genotype and to more genotypes or not? We hope it is. It gives us a path forward for more research, but again, rarely is that going to be something that's going to end up in your breeding program.
I agree.
Okay, thank you.
So I have a question to Alexandre. Potato is very difficult to manage, like to predict yield, as you said. So when exactly did you fly the drone to take all the indices to include in your kernel to predict the yield? And because we know that we can fly many dates after planting, but depending of the date, the correlation will be better or no. So what is the strategy that you use it to incorporate the fly drones information into your kernel?
Yes, basically during the entire growth, right? So the way the images are captured for the breeding programs in the Nordic region, they are very similar to all the countries that are part of this project we are working on. So it's from Denmark, Sweden, Norway, and Estonia, right? So they all have the same standards of flying the drones, because they use it for crop monitoring and to see if late blight will occur. So it's like in all the growing period. So I haven't tested like if sampling would impact the results we had, but in the end I just combined everything and get like the overall profile with area under curves. But maybe if we only consider part of it, it would be worse or not. It's worth it to try.
Okay, thank you, doctors, all for this engaging discussion. On behalf of the organizing committee, we sincerely appreciate your participation and for sharing your expertise with us today. As a token of your gratitude, we would like to offer each you a small gift.